finally, we're going to start talking about DNA viruses rather than those, silly RNA viruses. Who cares about those things? Another quick announcement. Those of you who have been following the syllabus have noticed that some of the subjects moved around a little bit. Today we're going to talk about polyoma and papillomaviruses. On Monday we'll talk about adenoviruses. And then on Wednesday I won't be here, but one of my graduate students will talk about the single-stranded DNA viruses, and these will be the eukaryotic single-stranded DNA viruses, and then also some really bizarre single-stranded DNA viruses that we're not actually quite sure what they infect, which is a really fascinating story. And then after that, we'll talk about herpes virus, pox viruses, and then these weirdo retroviruses, which are basically double-stranded DNA viruses, at least when they're sitting in the genome. So, and then finally finish up with viruses of archaea, the most important viruses ever, of course. And then also some of the giant viruses, which would be the most important viruses ever if it actually gave us some funding to study them, but we're working on that. So today, again, we'll talk about the small DNA viruses that are infecting animals, and those animals particularly being humans. But first, there are a couple of things I wanted to talk about as far as the exam is concerned. And yes, I do have your scantrons. In theory, I'm not supposed to give them back to you like I always do. Don't tell anybody. I'll keep doing that. And if there are some issues with scores showing up, etc., please let me know. Disconnects between what was on the scantron and so on and so forth. I've got scans of all of them as well. But there are a couple of things. In this one, again, I usually try and go through and find the questions that the most people miss, which means I did a crummy job of explaining it. This one blew me away because there were like 7% of the people who got it right, which means I did a really, really, really crummy job because that's like less than people could have guessed. So the question here is, the influenza virus hemagglutinin is cleaved by a blank protease and is a blank. That's not quite how the question was written, but that's the way the answers were. Viral protease, can it be a viral protease? Are there any viral proteases in influenza? No. Okay, so that leaves us with either not cleaved, but there's this fusion peptide, and you make fusion peptides by doing cleavage. So that leaves us with dimer or trimer as the two options. And evidently I didn't do a good enough job of mentioning that the vast majority of these receptor binding proteins are trimers, so we have 3 of them. The flavivirus is a real exception if they're dimers. So the correct answer is cellular and trimer. Again, maybe I totally spaced on including this in my lectures or told people it wasn't the case. I'm not quite sure why that is. So that was by far and away the question that most people had trouble with. Second one, which is much, much, much less, is only about 20%, so it's probably just guessing who people got this. Which of the following is required for flavivirus replication? And I will claim all of this is on Dr. Hirsch because he didn't do a good job of explaining it. But these are literally from my notes that I took when I was sitting in the back. I didn't even go back and look at his lecture about this. So interaction of the RDRP with a viral protein primer. What kind of viruses do that? Viral protein primers, VPG. I forgot everything because it was on the midterm last time, right? So I don't need to bother. Pico-RNA viruses or polyoviruses. And this chord is going to be the death of me yet. Covalent modification of the RDRP with ADP ribose. What the heck? Actually, turns out bacteriophage T4 does this, so I'll pick them up. Cellular DNA helicase. Why is there no way that it could be a cellular DNA helicase? These are RNA viruses. So that can't be right. Poly-A tail on the genome. Do these guys have poly-A tails? No. No, almost none of them do. And in fact, it's pretty rare that you've got poly-A tails on genomes. The pico-RNA viruses are really the exception rather than the rule as far as it's concerned. So even just by elimination, one in theory could have gotten to here. At least that's the way my crazed logic works as far as these kind of things. So any other questions on the exam? Again, the high score is 44. I'm going to scale from that. What I haven't done yet is put together the 2 highest scores yet. And if you look at the statistics, there's some clear outlier bumps, which are probably then going to get normalized away by the end of the course. So people are curious about that. Feel free to send me emails. Okay, so as I mentioned before, we're now at double-stranded DNA. So we've ignored now all these silly RNA viruses. Again, we'll get back to the double-stranded, so where are we? The single-stranded RNA going through DNA, finally getting the dsDNA here. One of the things, of course, which is really important about these double-stranded DNA viruses is that they're going to be functioning in a very, very cellular manner because cellular genome is double-stranded DNA. Replication is happening with double-stranded DNA, cellular replication. Cellular transcription is happening with double-stranded DNA. So a lot of these cases, it's going to be very, very similar in terms of what's going on cellularly and with these particular viruses. Yeah?
What's the difference between 6 and 1?
Oh, the 6 and 1 here. Now, the numbers here are this is just from our textbook, so the numbers are wrong. But these guys down here, these are the retroviruses, and these are all of the positive strand RNA viruses. And I just pull up this slide to sort of try and give some orientation to this. And I should have blocked out all these numbers because, again, they're wrong. So what about these DNA tumor viruses? And I was joking earlier. This is the reading for today. And it's this is part 2 of the molecular biology of tumor viruses. This is how people got really interested in studying these viruses. Turns out that anytime Stedman put quotes around things, what does it mean? Not really. The fact that these things can cause tumors in really extreme conditions is true, but generally not much of an issue in terms of cancerous growth. A couple of other things. Chromatin, we kind of mentioned this before. The genome of these polyomaviruses, and the papillomavirus is the same way, are packaged in nucleosomes made of cellular histones. So again, it's not just replication, it's not just transcription, it's even packaging of the genome, which is very similar to the host. In fact, a lot of what we know about genome packaging comes from studying these viruses. A lot of what we know about transcription comes from studying these viruses. A lot of what we know about replication comes from studying these viruses. And so that's why it's been also, first it was the tumor viruses that people were interested in, and then it just became this really great model for studying transcription and replication. A couple of key things here. T-antigen. The answer to any question on SV40 or polyomaviruses is T-antigen. At least that should be your default answer because basically it does everything. T-antigen stands for tumor antigen. And so when you actually did get tumors induced by infection with these kinds of viruses, the main protein that was being produced was this T-antigen, and could be detected through looking at antibodies. And so that's, again, horrible naming process, but the tumor antigen is that main protein. A couple of other things, and this has to do, again, mostly thinking about transcription. Divergent promoters, which should sound really, really familiar in terms of way back on that last midterm. That was Wednesday. Who cares? What other kinds of viruses have we talked about that have divergent promoters? Alpha, beta, gamma, lambda. And alternative splicing, which is also really important for these genomes. And we'll talk more and more about splicing as we keep going through the rest of these DNA viruses. Again, sort of classic outline, do very similar kinds of things. Where'd they come from? What's their structure look like? Finding entry, transcription. But the really big story here is replication. And to some extent, what I should put down here as well is regulation of the cell cycle. Because most cells that get infected by most viruses are not undergoing replication most of the time. And particularly for these smaller DNA viruses, they're so dependent on host replication machinery, they've got to stimulate the host to actually go into replication. And so regulation of cell cycle should be sort of an extra one, which goes in here. So where do these guys come from? Tumor viruses, sort of beating on this whole DNA tumor viruses. In fact, the first tumor viruses to be found were the RNA tumor viruses, interestingly enough. and those were all of the retroviruses. So this was the big surprise is, oh, we've got these viruses that are causing cancer that are not RNA viruses, and we'll get back to that a little bit later on. SV40, simian virus 40, was in the Salk polio vaccine and actually not completely inactivated in some cases. So many people, particularly those who are of a certain age, like me and older, who got the inactivated polio vaccine, actually were infected with SV40, seemed to cause them no trouble whatsoever. So how are they getting as a tumor virus? Well, it turns out that if you inject mostly rats, I don't think anyone's done this experiment with humans, probably ethically a real bit of a question, but really large amounts of these viruses, you can get tumor formation. And we'll see why that is. It's mostly because of the answer to all questions about polyomaviruses, T-antigen. So that's the reasoning. Actually, that's mostly having to do with regulation of the cell cycle. And that's what happens in most cancers, is you get dysregulation of the cell cycle. So it makes a lot of sense there. But most importantly, as far as I'm concerned, is really in terms of using these as tools to understand transcription, replication, etc., and also to some extent splicing. We've talked about the structure quite a bit already. Just wanted to go over this again very briefly. Very bizarre structures because they've got these nice five-fold axes of symmetry right here. There's a nice five-fold axis of symmetry because I can't draw a pentagon, but there are the 5 capsomers just outside of that. Everywhere else, there are 6. And so here's the one that's got 6 around it right here. But if you look at the middle of all these things, you can see they're very clearly star shapes, 5-pointed stars. So not stars of David, but 5-pointed stars. And so they're all pentamers, as opposed to quasi-equivalents where you have pentamers and hexamers. So that's the big difference here. Again, it's the, you can't have nice T numbers here, at least not in terms of T times 60 equals the number of subunits. It's because there are all 50 of them. And the other thing here is that they've got these beta barrel structures. Unlike the beta barrel structures, which we saw in all the picoRNA viruses, where they're sitting down, basically flat on the surface, these guys are sticking up. But beta barrel structures, very, very common in terms of the different structures of these particular virions. But again, we've talked about that already, so that's not as critical. On the other hand, the packaging of the genome is very different in these viruses, and the closely related papillomaviruses as well, in that the DNA is packaged in regular cellular core histones, H2A, H2B, H3, and H4, with the DNA wrapped around it. And if you're careful about purifying the genomes of any of these papilloma or polyomaviruses, you will see under the electron microscope that you have the beautiful beads on a string, where you get nice packaging of 140 odd nucleotides in terms of each of those wraps around. And so that's a great way of actually studying genome packaging, is to purify the DNA from these viruses. And you can get tons of DNA from these viruses, so it's a really nice way of doing that. Also, if you want to look at replication on a nucleosome-bound template, transcription on a nucleosome-bound template, this is a really good template to use for studying any of these things. So that's genome packaging. How do they get in? These, the polyomaviruses bind to sialic acid, which is also what the influenza viruses are binding to. What does this mean about hemagglutination? These also will hemagglutinate. So red blood cells also have sialic acid on the outside, so you can get cross-linking with these viruses. Unlike the case for, again, most of these viruses that we've talked about so far, they don't go in through receptor-mediated endocytosis. These are calveoli, a separate way of getting inside the cell. Once they get inside the cell, they escape from the calveoli. It's not entirely clear how that happens. People are still trying to figure that out. But then, and probably most importantly, is that these guys, in fact, the whole virion gets transported to the nucleus. Why is it getting transported to the nucleus? Yeah, because it's a DNA virus. It needs to use all of the cellular DNA replication, transcription, you name it, machinery. How that works is there's a nuclear localization signal, that's what NLS stands for, on the VP1. The VP1 is that major protein on the outside, and so you can see that it really is transporting a whole virion into the nucleus. Once it gets inside the nucleus, then the genome is released, and so there's a disassembly that happens inside the nucleus. And then you've got a nucleosome-bound template. You know, what better thing could you have for your RNA polymerases, your DNA polymerases, etc. So once you have your double-stranded DNA nucleosome-bound template, what happens? You get transcription, and this transcription is, again, from divergent promoters happening basically at one pace in a circular genome. Again, this should sound really, really, really familiar. Here, however, it's a little different because we're not talking about lysogeny versus lytic replication. This is actually basically a lot more like what's happening with lytic replication, only we have early genes and late genes here. The early genes, as usual, are going to be important for replication of your genome and regulation of what's going on inside the cell. Late genes are going to be what? Structural proteins, exactly. So the late ones we'll talk about a little bit later, but these are the pretty boring ones. VP2, VP3, VP1. VP1 is the vast majority. Again, that's what's making up all of those pentamers on the outside of the cell. Also has that nuclear localization signal on it. Oh, these are, sorry, forgot to mention this. So we even said these are small DNA viruses. Small just in terms of their size. They're only about 45 nanometers in diameter, each of those virions, but also small in terms of genome. Between 5,000 and 6,000 in terms of the polyomaviruses. So they're really pretty tiny in terms of their genome. It's not quite as small as some of the tiny bacterial viruses, but getting pretty close in terms of the sizes there. Because they've got such small genomes, they do what small DNA viruses of bacteria do, which is use multiple open reading frames. So same DNA, multiple different proteins that can be made from. And so you'll notice here the orange is representing coding sequence, and then these various different coding sequences which are overlapping with each other are now in different reading frames. How do they get different reading frames? It has to do with this splicing and differential splicing that takes place. It's also true for the VP proteins. You also have differential splicing. The other aspect of the genome here, which is really quite interesting, is that the transcripts actually overlap with each other. So your early transcripts overlap with the late transcripts. And what this means is you end up with double-stranded RNA here, and that double-stranded RNA is really important for regulation later on in infection. And we'll talk more about that when we actually get to that. Yeah, Peter.
Just the smallness of the genome, is that related to the fact that really all the machinery is already in the nucleus? It doesn't really have to code any of that?
Yeah, so
does that hold true for the rest of these?
Right. The question here basically is, since you've got this small genome, can you basically, you can get away with a small genome because the host has everything that you need. This is also true when we talked about some of those bacterial viruses. Is that they had, if you think about 5X174, basically it's using almost all cellular proteins, whereas we talked about some of the bigger ones like T7, which has its own RNA polymerase, it's got its own DNA polymerase, etc. So that sort of general idea is very similar. So you've got, when we talk about the larger DNA viruses that are infecting eukaryotes, those will also have a lot more of their own machinery. So in this case, very, very simplified things that they have. Yeah?
What's going on with all those vertical bars by PL?
What's going on with those vertical bars by PL? Great question. Let's move to the next slide. So, in fact, we're going to talk more about those a little bit later on. Those are all of the late promoters. And again, the eukaryotic virologist can't talk to the bacterial virologist. This is not left, it's late. So these are the late promoters, and then these are the early promoters right here. Late promoters, again, we'll talk a little bit more about these a little bit later on, but there are multiple different promoters which are used. Many of them don't have Tata boxes, which is really kind of bizarre. And those seem to be regulated mostly by T-antigen, exactly. And you'll also probably notice here large T-antigen binding. These are the binding sites to the large T-antigen, which are going to regulate expression of these various different promoters at PL, which again is going to give you your structural proteins. More importantly, as far as these early promoters are concerned, has to do with this enhancer site. And what are enhancers? Cellular enhancers. So way back when, like last term or wherever other time you took molecular biology. So they're DNA sequences which increase transcription. How?
Good processivity.
In affinity for, not for polymerases. Enhancers, you can move them upstream, you can move them downstream, you can flip them over, you can put them on different pieces of DNA. What happens to enhancers? You have incredibly creatively named enhancer binding proteins. So these are your, very often your transcriptional regulators. So these are the, yes?
Why are the Tata boxes on there if there's no Tata box?
So the Tata box, there is no Tata box for the PL promoters. Where's my pointer? But for the PE promoters, it's a perfectly normal Tata box. And this isn't a Tata box, it's an at box. No, that's when you're talking about going on this strand, which is where the PE promoter is going, that'll be your Tata box.
Okay.
So the early promoters do have a Tata box, and in fact it's regulated extremely similarly to most of your eukaryotic RNA polymerase II genes. And that regulation happens here through the activity of enhancer binding proteins, which bind to this enhancer, and you may or may not remember a protein called AP1, activating protein 1, which is the first transcriptional activator ever to be discovered. It was discovered because it binds to the enhancer of SV40P early genes. SP1, specificity factor 1, same thing. By binding to the enhancer of SV40 early promoters. And this enhancer, this is the one that they took and moved tens of thousands of base pairs upstream, downstream, flipped it over, put it in a different DNA molecule, and showed that it's still activated through DNA looping of all of those transcriptional regulators. So transcriptional regulators bind to the enhancer, interacting with components of the general transcription machinery through DNA looping in order to stimulate transcription. So that's how you're getting all of these. And again, it's cellular proteins. So it's not just the cellular polymerase, it's the cellular transcriptional regulators as well that are there. The other thing which you have in this intergenic region, again, between your early genes and your late genes, is the origin of replication, which is right here. Also, classic AT-rich sequence that's easily pulled apart by helicases. We'll get back to that a little bit later on. And gives you bidirectional replication, replication forks, etc. So let's look at these genes that are being expressed from this out-of-box control promoter relative to the enhancer binding proteins, and again, cellular enhancer binding proteins. These are the tumor antigens, I should say. This should be an S up here rather than just antigen. There are multiple different tumor antigens that are made from this early promoter. They're all identical at the N-terminus and different at their C-termini. And that's because of different splicing. And so these are 3 cases at the top, and we're going to actually ignore this 4th one down here. Again, incredibly creative naming, large T-antigen, medium T-antigen, small T-antigen. And we discovered one later that was smaller than the small one, so we had to name it something else and just called it the tiny T-antigen. So the main thing again here is that the N-termini are all identical, and you can see that, phi-glin pro, phi-glin pro, etc., phi-glin. And then it's differential splicing that gives you these different reading frames giving you different proteins. And this is differential splicing, again, not at all uncommon in terms of what you see in cellular splicing. Some splice sites are used, other splice sites are not, etc. And so it's that differential splicing which gives you different proteins and different open reading frames that do that. Okay, happy, unhappy way of using a relatively small amount of genome to give you most diversity of sequences. Well, before we talk about what these things do, we can do our clicker question for today. First one anyway. There will be a second one later if we get there. Distribution of mRNA coding sequences in polyomaviruses is most similar to mRNA coding in Ebola, measles, SARS, 5174, and Lambda. I'll review, yes. Old stuff, I forgot that. That was like midterm stuff. Again, feel free to discuss. Can 5, 4, 3, 2, 1. Unfortunately, I don't agree. I think it's much more like what you see in 5174. And the reason is we're using multiple different open reading frames. 5174 also is using positive and negative strands. It was so long ago, exactly. But these are the kinds of questions that Stedman is not unlikely to use later on. But of course, as we normalize, most of this is going to disappear, except for the few people who either got lucky or are crazy enough to think like I do. Okay. Okay, let's talk a little bit about what these different antigens. Yeah.
Oh, does the fact that the chromosome is circular add complications to that histone wrapping that our linear chromosomes don't, or it doesn't matter?
Oh, so the question here is, sorry to repeat just so it's recorded here. Does the fact that it's a small circular genome cause problems in terms of nucleosome binding as opposed to having ends which stick out? This has to do with supercoiling, which they discuss a little bit in the chapter in the textbook. And so it turns out that there's only a certain number of nucleosomes that can be bound to that particular piece of DNA. And so you can't get to a much higher amount of nucleosomes in that case because it's constrained in terms of the supercoiling. If you extract all the nucleosomes, all the histones, then you get massive wrapping of the DNA around itself because, again, it's covalently linked. If you had a linear genome, that would be a lot less of a problem.
So that must constrain the size of these genomes of these kind of viruses.
Well, they can only get to a certain small size, relatively speaking. Yeah, and so and it turns out the nucleosome packing is slightly different in these genomes than you would see in a cellular chromatin. In fact, the nucleosomes move around a lot more in cellular chromatin than they do on these templates. Okay, other questions before we start talking about what all these crazy differentially spliced things do? The answer that I didn't have on that question was also influenza virus, possible answer for the clicker questions, because they also are doing differential splicing. So, and there's no splicing in lambda. And we'll talk much more about splicing as we move further on again through the rest of the course. But yeah, what are these antigens doing? Basically, they're involved in either standard early functions, which are going to be genome replication, or modifying something that's happening with the host. And we talked about this way back when. I know we're rolling back the tapes. Bacteriophage T7. What happened in bacteriophage T7 right after you got infection? You shut down all the restriction endonucleases and you shut down cellular RNA polymerase. Again, because you had all these viral proteins that did that, but it's regulation of things that are going on inside the cell. Exactly the same thing is happening here for basically two reasons. The main one is that most cells aren't normally undergoing replication. If they're aren't undergoing replication, then you're not going to have all of the dNTPs, you're not going to have all the enzymes, et cetera, that you need to make more DNA. Well, if you're a virus, again, to totally over-anthropomorphize here, you need these things to make more of the DNA. So you need to turn on the cell cycle. And so the small T-antigen is mostly involved in that. And what the small T-antigen does is it activates the MAP kinase cascade. And so a couple people asked me, what about cell biology in terms of this course is concerned? It's one of those cell biology things. So go back and take a look at that. MAP kinases are really important in terms of activating the cell cycle, mitogen-activated protein kinases. There's this whole cascade of phosphatases of phosphatases of phosphatases and kinases of kinases of kinases. We're going to ignore them and just look at the MAP kinase cascade, which eventually leads to activation of activation protein 1, which binds to various enhancers in the genome, turning on cyclin-dependent kinases, activating the cell cycle. This is usually downregulated by phosphatase, and particularly this protein phosphatase 2A. Protein phosphatase 2A, what does it do? It takes the phosphate off of this MAP kinase, gives you inactive, and does not stimulate the cell cycle. If you block the protease, you end up with more phosphorylated MAP kinases that end up giving you progression in the cell cycle. So it's blocking the phosphatase, and in blocking the phosphatase, you end up with more activation of the cell cycle in this case. AP1 is also involved in transcriptional regulation of the virus genome. There's the middle T-antigen, again, medium-sized T-antigen. People use these interdependently, but what this middle T-antigen does is basically tricks the cell into thinking that, hey, we've got a active tyrosine kinase. The best known of these tyrosine kinases is called C-SARC. We may get a chance to talk about that when we talk about retroviruses later on, the RNA tumor viruses. That's how these were found. But here we have tyrosine kinases, which phosphorylate on tyrosines. Once you phosphorylate on tyrosines, you turn on the MAP kinase cascade. And so this is basically another way of stimulating the MAP kinase cascade, and this is a positive regulation as opposed to the removal of negative regulation. So those are the small and the middle. The guy that does most of the work is the large T-antigen. This, again, generated by differential splicing, again, from the early promoters. This has an absolutely amazing number of different domains which do all kinds of different weird and wonderful things here. What I wanted to start out with is talking about the interactions, which are up on top here of the screen. All of these things are on the top. These are different regions of the large T-antigen, what it interacts with. The retinoblastoma protein, which we'll talk about a lot more as we move through. This is their classic tumor suppressor protein. You lack it, you get tumors, and very important for progression through the cell cycle. We also have The very first ever characterized nuclear localization signal is from the large T-antigen. So specific stretch of amino acids, which interacts with all of the nuclear import machinery. So it's imported in the nucleus. Various different domains present inside the protein. This one, the J domain, is the DNA J-like domain, important for protein folding and assembly. Yeah.
So is it making retinoblastoma protein, or is it interacting?
It's just interacting with that.
Okay.
So the stuff on top here is what it's interacting with, and down here are the different domains and what they do. So in addition to interacting with RB, the large T-antigen also interacts with P53, the guardian of the genome, etc. Anyone know how P53 was originally discovered? It's a protein that interacts with the large T-antigen that's 53 kilodaltons in size. So again, SV40 and large T-antigen is really sort of the nexus of thinking about so many different things, about molecular biology, cancer, etc. There are other things that large T-antigen interacts with. P300, anybody know what P300 is? Histone acetyltransferase, important for regulation of transcription. Histone acetyltransferases do what? Pull histones off of DNA. Why would you want to do that? You can get more transcription, particularly at promoters that don't have Tata boxes, like the late promoters in SV40. So it's also serving as a transcriptional regulator. And you can also see down the bottom here this transactivation domain. So transactivation is classic transcriptional activation. And how does it do that? Interactions with P300 here. Other interactions, DNA polymerase alpha, Tata binding protein, again, DNA polymerase alpha, topoisomerases. And again, this basically tells you it does everything. This is your Swiss Army knife, or actually where in Portland, this is your Leatherman tool, in terms of interacting with everything else which is there. The large T-antigen structure has been solved. It does lots of different things. Again, yeah, David.
To what extent should we be filing this all under SV40 versus polyomaviruses in general?
So in terms of, I am probably going to use, and this is bad from my point of view, polyomaviruses and SV40 more or less interchangeably. So I'll try and be specific on, say 3 weeks from now, June 8th, I think it is. But I'll try and be specific about, which one I'm talking about there. But basically, as far as we're concerned for this course, SV40 and polyomaviruses can be used interchangeably with each other. Yeah, Lou?
So you said BP1 has an NLS.
Yep.
That's bringing the capsid into the nucleus
Yep.
and the genome is released. So why does the large T-antigen need an NLS?
Why does the large T-antigen need an NLS?
Just to stay there?
What happens in the nucleus? You have transcription, right? Where do mRNAs go? Out into the cytoplasm. They get translated. Where do you need large T-antigen? Back in the nucleus. So that's what it's about. Yeah. So it's after it's been made and then going back into the nucleus. Now, A, for all the transcriptional regulatory stuff, but also leading into this, thank you, for replication purposes. And so this is the structure of actually just part of the large T-antigen. Back up real quickly. It's this DNA helicase domain down here. DNA helicase should tell you what it does. Oh no, we're going the wrong direction here. Sorry about that. What DNA helicases do is they just pump DNA and literally pull strands apart. This is exactly what the large T-antigen does. Serves as a replicative DNA helicase for SV40. And just like all helicases, it burns through ATP. It also interacts with RPA, which is what? Wow, this is like even before the first midterm in this class. RPA is replication protein A. It's a Monday morning. I had too many exams. No, it's not Monday. It's Friday. It's almost the weekend.
Oh my god, I please.
Is single-stranded binding protein. Single-stranded binding proteins, once you have the two strands separated, you need the single-stranded binding proteins in order to make sure those two strands don't go back together. DNA Pol alpha does what?
Oh, yes.
It polymerizes DNA. Yeah, thanks. What part of DNA replication is Pol alpha involved in? Making primers. So it's not the primase, it's what extends the primers. And topo 1, what does DNA topoisomerase 1 do?
Reduce binding.
Cuts and allows supercoiling, which gets back to David's question about these small genomes and dealing with what's happening there. So DNA replication, this should look really, really, really familiar, hopefully, although no one seems to remember what happened last term. This is formation of helicase, interaction with topoisomerase 1. You have Pol alpha, which eventually is then going to move over to Pol delta, Pol epsilon. Replication protein A, binding to your single-stranded DNA. This is your classic replication fork. And this is, in fact, how, in some cases, even in molecular biology textbooks, they label the replicative helicase as SB40 large T, which is really kind of crazy because, but it took a rather long time to figure out what the cellular DNA helicase was that was involved in replication. Turns out it's the mini chromosome maintenance proteins, MCM proteins. But for a long time, this was the only replicative helicase that people knew about, was the SB40. And otherwise, again, replication completely normal in terms of everything else as far as the rest of the genome is concerned, but completely dependent on the helicase. And again, that's what's true with all DNA replication. The main thing you do is get those two strands apart. Once the two strands are apart, you're in good shape. But it's getting those two strands apart, which is the really critical step. We talked about the late transcripts quite a bit already. Main thing here is we're stimulating this based on the presence of the large T-antigen, which binds over here on this side. The binding of the large T-antigen, I should have brought up the intergenic sequence here as well. Binding of large T-antigen also blocks expression from the early promoter, so it's a nice self-regulatory mechanism. Another self-regulatory mechanism is that these transcripts down here, the very end of your late transcripts, form double-stranded RNA with the transcripts from E. Double-stranded RNA leads to degradation of those transcripts. So it will give you degradation of those processes. There are also cellular regulators that are involved in this process. But it's mostly, again, the important stuff as far as we're concerned is really going on with the early promoter. How do you get assembly? Assembly happens, not surprisingly, in the nucleus. The capsid comes into the nucleus due to nuclear localization signals on the major capsid proteins. That all gets assembled in the nucleus, and then the whole thing gets transported back out. So that's the main process, yeah.
The capsid proteins come in, or the entire capsid?
The entire capsid comes in, and the entire capsid then gets transported back out. So the assembly happens in the nucleus.
The DNA goes in and it goes back out?
So the DNA, so when you get infection and the capsid comes in, one of these polyomavirus capsids comes in, you have disassembly of those capsid proteins in the nucleus, and you have your double-stranded DNA associated with nucleosomes. Then you have replication of that double-stranded DNA through the regular, bidirectional process here, and then binds to nucleosomes, gets packaged in the nucleus as a virion, and that virion gets transported back out. So it's the virion. The only thing that the cytoplasm sees, as far as these viruses are concerned, is just virions. So virion comes in, virion goes back out. So it's all. And if you look at the nucleus of an infected cell, you'll see full virions of these polyomaviruses, and also for the papillomaviruses, as we'll get to eventually. Yeah.
Quick question.
Yeah, sure.
So
No worries.
they're in the virion capsid, and that's kind of like a protection mechanism. So I'm trying to envision what the benefit is, for.
Why be in a capsid when you go back out? Well, again, no. The simple, quick, easy questions end up being, that's fine. Turns out that double-stranded DNA in the cytoplasm is a major defense mechanism that cells use to say, hey, wait a minute, we got this virus in here, this is bad.
Right.
And so it's a way, it's not so much a degradation thing, but there's a signaling that happens with double-stranded DNA, free double-stranded DNA in the cytoplasm. So it's DNA which is in the wrong place. And again, to totally over-anthropomorphize the cell then knows that there's a virus which is there. Because if there's double-stranded DNA in the cytoplasm, something's wrong. And so there are mechanisms to deal with that.
It's like stealth mode.
Yeah, so basically being in the very honest stealth mode. I was getting back out. Yeah.
They, so where did the virus get out? Do they find out, or do they ice out?
Oh, so the question is how do these virions actually get out? The answer to that question is I'm not completely sure, but these are not enveloped viruses. These are naked viruses. So somehow they're being released. I don't think it's a burst process, but I could be wrong on that. I'll take a look. Actually, it's a great question. I was thinking about it as I put the lecture together. How are they actually getting out? Okay, so just briefly talk a little bit more about tumor viruses. Again, polyomaviruses usually don't cause any kind of cancerous growth, and this is, David, getting back to your question about SV40 and polyomaviruses going together. This is where SV40 diverges, because there are actually a couple of these polyomaviruses which do actually seem to cause some cancerous growth, and the reason for that is, not surprisingly, the small T-antigen, the medium T-antigen, what are they doing? They're stimulating the cell cycle. And to some extent, the large T-antigen, a little bit less so as well. So, not surprisingly, stimulating the cell cycle can lead to this cancerous growth. Turns out it's very rare that this happens. 1 in 100,000 or something, way less than that. And what happens there is this only happens in what are called non-permissive cells. And we haven't really talked too much about permissive cells or non-permissive cells. Basically, a permissive cell is one where you can get virus replication to take place. You have all the machinery that you need. A non-permissive cell, you don't have all of the machinery that you need for that virus to replicate. And so what seems to happen is that in these non-permissive cells, the virus is not replicating and getting out, but still all of these T-antigens are being made and stimulating the cell cycle. And so that ends up leading to cancerous growth. It's a relatively rare process. There are some other polyomaviruses which have been associated with disease, and this is, in fact, getting back to the chapter here. Merkel cell polyoma virus, MCPyV, isolated from many Merkel cell carcinomas, but not all of them. There's this whole link about it here, but the basic take-home message, and this is from a couple of years ago now, impact of positivity on the course of disease is controversially discussed. Highly controversial whether this is an associated virus or an actually causal virus in terms of these particular diseases. Okay, any more questions on the polyomaviruses? We'll talk about papillomaviruses, but molecularly they're extremely similar to the polyomaviruses, so that's why I'm lumping them together. With one big exception, and that is these really are tumor viruses, as opposed to the polyomaviruses, which are only going to be causing tumors at these much, much, much higher levels, and much more rarely. These viruses, in fact, not unlike what happens with the polyomaviruses, they're replicating in differentiated cells, and particularly in skin cells, and there are a number of vaccines that we may or may not have a chance to get to at the end here. This should look really, really familiar in terms of what we're talking about here. The main difference here has to do with transcription relative to what's happening in the polyomaviruses, and also the different proteins that are there. Where do people find the papillomaviruses? Mostly warts, plantar warts, etc. These are papillomaviruses. Really don't seem to be terribly detrimental, maybe disfiguring, but there's lots and lots of these around. There are literally, I think, hundreds. I just read a paper, I think there are 480 some new different papillomaviruses which they just sequenced. So there are ridiculous numbers of papillomaviruses out there. They are quite infectious, and again, having to do with how they're being released, in fact. Most of these are benign, quote unquote, Whether they're beneficial is another interesting. But in some cases, there's clearly a direct connection between some of these papillomaviruses and cervical cancer and any of these, or a number of squamous cell carcinomas that are involved in that. So this is the main reason that people have been interested in these particular viruses and the differences between those and the polyomaviruses. And in fact, for a very long time, this was highly controversial because, in fact, people had gone from, oh, these are DNA viruses causing cancer originally with like SV40 and these things, but oh, no, they don't really cause cancer at all. And then Harald Sohausen, whose picture's right at the end that we're not going to get to probably, who got the Nobel Prize, I think, 2 or 3 years ago, kept beating on the fact that some of these papillomaviruses really did cause cervical cancer, which led to the development of these vaccines. So it was sort of this, oh, look, viruses cause cancer. No, viruses don't cause cancer. Some viruses actually do cause cancer. And so that's where these papillomaviruses come from. How do they get in? They're not binding to sialic acid, so they're not causing hemagglutination. These guys come in through epithelial cells, and again, mostly things like skin epithelia. Different proteoglycans come in through regular endocytosis processes and express early genes, not surprisingly. This is a circular double-stranded DNA. Unfortunately, it's shown here as a linear form. There are regulatory sequences here. This is not like you have in SV40, which is where you have divergent promoters. These are actually overlapping promoters, early promoters and late promoters. Got a whole bunch of early genes and a couple of late genes. What do the late genes do? They make structural proteins. What do early genes do? Help replicate the genome and regulate the host. So the late genes are pretty straightforward. They mostly seem to be dependent on the differentiation of different host cells. So differentiation is this process of sort of maturation of cells. You only get production of these late protein genes in differentiated cells, and particularly here when we're looking at skin cells, right here in these spinous layers of epithelia. So your HPV or any of these papillomaviruses, they're still going to be being produced here, and particularly, again, produced in these differentiated cells. All of these are just like you see with the SV40 structure, hexamers fitting into, pentamers fitting into hexameric holes. The big difference here has to do with the actual proteins, and particularly E1 and E2, early protein 1 and early protein 2. You can kind of think of E1 and E2 as being a combination of what you have in the large T-antigen, and this is the way I like to think about it. So E1 is a DNA helicase, binds to replication origin, initiates DNA replication. But that seems to be pretty much the only thing that it really does, relative to large T, which is doing absolutely everything. E2 does all the other things that large T antigen does, binds to the replication origin, is a transcriptional enhancer, and also involved in changing a little bit of the cell cycle. Most importantly, in terms of cell cycle, E6 and E7 here, which we'll look at in a little bit more detail these next couple of slides. E7, just like the large T antigen, interacts with RB. Just need to back up a little bit and talk about what RB normally does. So normal RB function is down here. Retinoblastoma protein binds to the E2 factors. It's ridiculously confusing here. Sorry about this. Again, it wasn't my choice. E2 factors are cellular transcriptional regulators. So it has nothing to do with the papillomavirus E2 protein, which is the helicase. So these are cellular transcriptional regulators. These E2 E2 factors will bind to RB, and once they're bound to RB, they're not going to stimulate the cell cycle. So RB is serving as the tumor suppressor. Again, you're missing RB. You end up these E2Fs expressing all of these cell cycle proteins. In the normal cell cycle, you have cyclin-dependent kinases that are going to phosphorylate RB. Once you phosphorylated RB, E2F is available to go ahead and transcribe, or actually there are lots of E2Fs, transcribe all these things that you need for the cell cycle. So this is normal cell cycle processes. P53 will normally lead to blocking of this progression in the cell cycle. What E7 in the papillomaviruses does is it binds to RB basically like phosphorylation would happen, and so stimulates this progression of the cell cycle. And that's kind of shown up here. HPV, the E7 protein, is interacting with RB. We'll talk about E1A on Monday morning. SV40 large T-antigen interacts with RB, and SV40 large T-antigen also interacts with P53, and now we have that other early protein from HPV, which is the E6 protein. That interacts with P53. P53, guardian of the genome, etc. But P53 is a really interesting protein. First found was binding to large T-antigen and SV40. But it's involved in both cell cycle arrest, but also stimulation of apoptosis. So it actually has these sort of two different roles that it plays. Apoptosis, of course, is programmed cell death. If things are going really, really bad for one cell, it commits suicide to save the rest of the organism. Again, total oversimplification there. But that's if going is getting really, really bad. If the going is just getting slightly bad, and you want to repair your genome for some particular reason, oxidative stress, you name it, then what happens is P53 will lead to cell cycle arrest and allow the cell to repair itself. And so it's a sort of fine line between going all the way to apoptosis or hanging out for a while and hoping that things are going to get better. So here, the E6 protein interacts with, again, another incredibly wonderfully named protein, E6AP, activating protein, which is a cellular protein that then leads to degradation of P53. If you degrade P53, P53 can no longer induce apoptosis or block the cell cycle, which is exactly what you need in terms of papillomavirus replication, because it's got to go through the cell cycle in terms of making all of these appropriate genes, and you don't want them to undergo apoptosis, because if it undergoes apoptosis, all the virions which are inside the cell are going to croak just with the rest of the cell processes as well. Okay, he's pausing. That means he's just about to ask a clicker question, right? In terms of DNA replication, which of the following papillomavirus proteins is most similar in function to SV40 large T-antigen? 1, 2, 6, L1, or L2? Beluga whale, I forgot. By definition. got a pretty even distribution this time.
Just structural.
Yeah.
It's concerning.
Yeah, E1, E2 are like telophase related.
Okay, we're divided on this one. Key to this particular question is what?
DNA replication.
DNA replication. DNA replication's about what? Pulling the two strands apart. What does that? It's the helicase activity. And helicase activity is in
E1.
E1. Okay, I'm just going to finish up with these last two slides. We're not, unfortunately, going to get a chance to talk about the vaccines, but I did want to talk about sort of why the papillomaviruses do seem to be cancer-causing as opposed to what happens with the polyomaviruses. And the important thing here seems to be having to do with those interactions, separate interactions with P53 and RB, because you've got E6 and E7 in these processes, as opposed to just stimulating the cell cycle process, which is what you have with the T-antigens that happen in the polyomaviruses. P53 leads to activation of P21, and that then was going to block PRB activation. So we've got this difference, again, between E6 and E7 relative to what's happening with the large T-antigen. Again, same thing, however, you see with your massive amounts of the polyomaviruses, where you have only this happening in non-permissive cells. The same thing is true here, is that the transformed cells, i.e. cancerous development, are incompetent for replicating the virus. And so it's a complete difference here. Producing virus, you're not going to be making cancer. If, however, you're not producing virus, you have a chance of making cancer. And in cervical cancer, it's really E6 and E7, again, interacting with PRB and P53. Everyone's favorite cancer here, HeLa, all the HeLa cells, are in fact because of transformation with this papillomavirus E6 and E7, and they're wonderful in terms of growth. They're not good in terms of HPV disease. It's not just cervical cancer, anal cancer as well. Probably 70-odd million people in the U.S. are infected, and any sexually active person is infected with an HPV, and probably, certain, 10 to 20,000. And one of the important things here is many men, and a lot of people sort of ignore that when they say HPV. They always think cervical cancer, and we aren't, us males have cervixes. But yeah, there's a lot of other cases here. Almost all of these are HPV 16 and HPV 18. I mentioned there are huge numbers of these. These are what are being vaccinated. And in the actual vaccine, which we'll see the picture of right here, Gardasil, those of you who are parents, males, or male children or female children, vaccinate, vaccinate, vaccinate. So with that, vaccinate, vaccinate, vaccinate. It's the weekend. I'll see you on Monday.